Selamat siang. Kita akan belajar tentang basic konsep fungal pathogenesis. Mikologi kedokteran merupakan cabang biologi yang mempelajari jamur, fungus atau fungi, dan penyakit yang ditimbulkan oleh jamur. Konsep dasarnya, jamur bersifat eukariotik, ukurannya sekitar 4 mikron, heterotropik, aerobik, tidak berklorofil, jadi tidak bisa mensintesa makanan sendiri. Hidupnya saprofit atau parasitik. Morfologinya, jamur ada dalam bentuk ragi, mol, atau dimorfik fungus. Ragi bertunas, misalnya akan dialbikan. Mol atau kapang dalam bahasa Indonesia berbentuk IPA atau benang-benang jamur, misalnya Fungal. Ada jamur yang bersifat dimorfik, hidupnya sebagai ragi di dalam badan atau dalam kudu pada 33 derajat Celcius, dan sebagai mul di dalam bebas pada suhu 25 derajat Celcius. Jadi ada ragi, mul, juga fungus. Struktur dari jamur, dinding sel mengandung lapisan kikrin, beta-glukan, mangan. Di dalamnya ada plasma membran mengandung ergosterol, berbeda dengan bakteri. Kalau bakteri itu kolesterol. Mengandung protein, lipid. Pengobatan jamur memakai amphotericin, tempat kerjanya pada plasma membran ini. Di dalamnya ada sitosol di sitoplasma, mengandung mitokondria, inti, endoplasmik retikulum. IPA itu ada yang bersepta dan satu inti pada tiap septa, ada yang tidak bersepta atau koinositik, yang banyak inti. Jadi intinya bisa bergerak bebas di dalam sitosol. Unit reproduksi dari jamur ada conidia dan spora. Conidia itu bersifat aseksual, ada yang makroconidia, terdiri dari banyak sel. Mikroconidia bersifat uniseluler. Spora bersifat aseksual, dibentuk dalam sporangium, disebut sebagai sporangiospora. Misalnya pada Rhizopus. Spora yang seksual terjadi karena penggabungan hipa, misalnya ascospora dibentuk dalam ascus, basidiospora dibentuk dalam basidium. Spora dan zygosspora terjadi karena penggabungan dua hipa. Pada zygosspora hipanya sama, pada oospora penggabungan hipa yang berbeda. Jadi ini reproduksinya conidia dan spora, dan itu ada di mana-mana di alam, bebas di udara. Ini contoh blastoconidia pada aspergillus, pada candida, kemudian pada gambar B blastosporanya lepas menjadi sel ragi itu sendiri. Pada yang C terjadi blastospora yang menyambung menjadi pseudohipa. Pada D ada hipa yang koinositik dan tidak bersepta, idenya bisa bergerak lebih leluasa. Nomor E dan F merupakan hipa bersepta. Jadi satu septa ada satu inti. Ini contoh yang lain. A itu ada blastoconidia, B pada chlamydospora, ada yang di dalam hipa, ada yang lateral, ada yang terminal chlamydospora. Pada gambar C terjadi arthrospora karena fermentasi dari hipa. Hipanya patah-patah, kemudian dindingnya menembang menjadi arthrospora. Pada gambar D ada sporangium, di dalam terdapat sporangia spora. Pada E di gambar aspergillus dengan microponidia. F ada microponidia dan macroponidia. Yang macroponidia terdiri dari banyak sel. Klasifikasi daripada jamur berdasarkan unit reproduksinya dibedakan yang aseksual dan yang seksual. Yang aseksual istilahnya anamorf, yang seksual istilahnya teleomorf. Dari asal agennya ada yang endogenous, asal dari badan sendiri, misalnya pada infeksi opportunistik. Candida itu ada normal dalam badan. Ada yang exogenous dari luar. Berdasarkan patogenitasnya, ada yang primer patogen, ini bukan infeksi endemik di wilayah tertentu. Yang opportunistik patogen, ada kekebalan yang menurun, sehingga yang normal menjadi patogen. Flora normalnya menjadi patogen dan yang opportunistik. Dari epidemiologi dibedakan yang nosokomial, misalnya orang yang dirawat di rumah sakit karena kekebalan menurun menderita penyakit baru di rumah sakit. Community acquired artinya didapat di daerah yang endemis, tergantung wilayahnya. Itu diberikan perbedaan antara jamur dengan bakteri. Jamur mempunyai ukuran lebih besar, sekitar 4 mikron. Berinci dalam sitoplasma ada mitokondria. Sel membran mengandung ergosterol. Dinding sel terdiri dari labenkitin. Sporanya seksual. Ada thermal dimorfisme, jadi ada yang bentuk ragi, ada bentuk hipa, tergantung pada pH, pada kondisi yang lain-lain juga. CO2 juga berpengaruh. Dinding sel luarnya terdiri dari kitin, sehingga obat-obat yang dipakai untuk pengobatan bakteria tidak akan mempan untuk obat-obat jamur karena lapisan berbeda. Karena bakteria terdiri dari peptida dan glikan. Peran dari jamur sangat banyak. Antara lain sebagai program normal di badan, misalnya Candida albicans hidup di mulut saluran cerna kelamin perempuan. Kemudian jamur juga berperan sebagai dekomposer bersama bakteri menghancurkan sisa makanan, sisa tanaman, sisa binatang yang mati. Kemudian jamur juga bermanfaat dalam produksi antibiotik, misalnya penisilin berasal dari penicillium, juga bermanfaat untuk produksi minuman, alkohol misalnya. Mushroom sebagai makanan juga merupakan jamur. Banyak sekali spesies jamur yang dikenal, tapi sedikit yang patogen. Tapi semuanya bisa bersifat oportunistik patogen, tergantung daya tahan daripada orang-orang yang bersangkutan. Efek dari bumi terhadap manusia adalah mikrotoksikosis karena makan jamur secara tidak sengaja, misalnya mengandung aflatoksin, itu bersifat karsinogenik, menimbulkan kanker hati, bisa menimbulkan hipersensitif, misalnya rinitis, asma bronkial, misalnya pada Aspergillus. AC itu mestinya dibersihkan setiap 6 bulan minimal karena merupakan sumber daripada Aspergillus. Menimbulkan kolonisasi dan penyakit. Manusia punya barrier yang baik terhadap infeksi jamur, misalnya kulit, mukosa, dan sekresinya. Namun infeksi bisa terjadi karena rusaknya barrier, karena trauma atau penurunan imunitas pada DNA. AIDS, orang dengan debil, sering terjadi kandidasi pada sudut mulut pada penderita dengan AIDS. Jadi yang kena mucocutaneous. Fungal pathogenesis barrier pada manusia berupa kulit, mukosa, flora normal, saliva, silia. Di orang sehat punya kekebalan alamiah terhadap infeksi jamur, walaupun selalu kontak dengan propagul. Propagul itu alat reproduksi dari jamur, bisa berupa spora atau conidia. Mikosis berkaitan dengan status imunitas host dan paparan lingkungan dibanding dengan infeksi. Infeksi terjadi karena kecelakaan, misalnya trauma. Barrier fisiologis terutama untuk tumbuhnya jamur di dalam badan, ada temperatur dan ada dose potensial. temperatur tubuh kita 37 derajat Celcius tidak cocok untuk perkembangan jamur. Redopotensia itu artinya reaksi oksidasi reduksi. Oksidasi itu menghasilkan tenaga, reduksi mendapatkan tenaga. Bila jamur mampu beradaptasi dan mengatasi barrier jaringan, maka infeksi akan terjadi dan karakteristik jamur akan berubah. Misalnya pertumbuhannya menjadi lebih cepat. Penyakit jamur ini tampaknya sebagai reaksi atau fenomenon karena kecelakaan. Ini jamur bukan menyerang manusia, serangan terjadi karena jamur kecelakaan. Jadi bukan untuk menurunkan diseminasi spesies, kecuali dermatophyta. Kecelakaan misalnya karena duri bunga mawar yang mengandung jamur menimbulkan mikosis subcutis. Jarang jamurnya menyerang kecuali ada kecelakaan. Mekanisme dasar pathogenesis jamur adalah kemampuannya untuk beradaptasi terhadap lingkungan jaringan, yaitu suhu 37 derajat Celcius, dan bertahap terhadap dialisis oleh pertahanan seluler dari tubuh. Ada sel T yang menyerang. Jadi kalau jamur bisa mengatasi itu, maka infeksi akan terjadi. Jamur mempunyai preferensi yang suka menyerang manusia di golongan antropofilik, misalnya Trichopiton rubrum. Biasanya sering terjadi serangan tinea corporis di daerah ikat pinggang, biasanya pada bagian ikat pinggang yang keras, sering di sana terjadi oleh Trichopiton rubrum. Yang zoofilik, jamur suka menyerang binatang, misalnya Microsporum canis, Trichopiton perucosum pada kuda. Geofilik, jamur yang hidup di tanah, Microsporum dipsium. Jamur yang zoofilik dan geofilik biasanya bersifat akut, disertai infeksi bakteria dan lebih mudah untuk disembuhkan. Sedangkan yang antropofilik biasanya infeksi yang kronis, lebih susah untuk disembuhkan. Lingkungan bisa memudahkan infeksi jamur, misalnya kita ada di daerah tropis, panas dan lembab. Karena itu hindari pemakaian pakaian nilon atau pakaian ketat. Karena daerah yang ditutup akan panas dan lembab, memudahkan infeksi kandida. Habit atau kebiasaan, terutama infeksi kronis. Dulu tukang binatu itu mencuci pakaian biasanya kena serangan kandidiasis pada kuku. Karena dampak mesin itu berkurang prevalensinya. Perubahan floral normal, misalnya pemakaian antibiotika dosis lama dan tinggi, akan merusak flora normal sehingga terjadi kandidiasis. Pasien debil, artinya pasien dengan keadaan yang menurun pada pemakaian sitotoksik, obat anti kanker, pemakaian X-ray, juga pemakaian terapi steroid menurunkan daya tahan. Kekebalan yang menurun pada AIDS juga memudahkan terjadinya infeksi. Opportunistic mycosis karena gangguan daripada flora normal, itu terutama pada gangguan imunitas juga. Penyakit jamur dikenal dengan nama mycosis. Dibedakan menjadi superficial, termasuk panu, tinja versicolor, tidak sakit, rasa gatal terutama berkeringat, sering karena kosmetik tidak baik. Jadi orang berobat bukan karena sakit, tetapi karena kosmetik. Mekanisme terjadinya gatal belum jelas. Dermatophytosis menyerang epidermis disebabkan oleh jamur dermatophyta, termasuk Trichopiton, Microsporum, dan Epidermophyton yang merusak jaringan dengan keratinase. Infeksi biasanya kontak langsung dengan penderita. Bagaimana pathogenesisnya belum diketahui dengan baik. Mycosis subcutaneous termasuk sclerotigosis biasanya menyerang saluran dan telinga kita bening. Chromomycosis, mycetoma biasanya pada kaki dan sulit sembuh. Infeksi yang terjadi karena trauma pathogenesis mekanismenya belum diketahui dengan baik. Yang sistemik jadi karena inhalasi disebabkan oleh jamur dimorfik. Jadi jamur dimorfik itu di alam sebagai HIPA dia, kemudian dia masuk dalam badan menjadi ragi. HIPA yang dihirup inhalasi sampai ke saluran pernapasan bawah akan ditangkap oleh makrofa pada alveoli yang biasanya pasien sembuh. Apabila tidak sembuh, maka jamur ikut sirkulasi bersama makrofa, ikut sirkulasi sistemik menuju ke organ-organ. Itu yang sistemik mycosis. Yang opportunistic mycosis, semua jamur ada tendensi untuk opportunistic patogen pada AIDS, pada DM, pemeriksaan jamur biasanya memakai KOH 10%, bersifat keratinolitik, merusak keratin pada kerukan kulit yang ditaruh di objek gelas, diisi KOH 10%, kemudian ditutup dengan cover gelas. Akan tampak HIPA dan spora. Jadi yang diketahui adalah dengan KOH bahwa ada infeksi jamur. Spesiesnya tidak bisa ditentukan. Untuk spesies diperlukan kultur yang bisa dipakai antara media saburu dan dextrose agar. SDA. Sering disadari antibiotika untuk mencegah pertumbuhan bakteri, ditambahkan siklohexamid untuk mencegah kontaminan jamur. Jadi yang ditumbuhkan adalah jamur yang berasal dari kerokan kulit. Selain kerokan kulit tentu ada juga dari sekutum, tergantung jenis penyakitnya. Sebagai resume, humus bersifat eukariotik, heterotropik, tanpa klorofil. Propagules merupakan alat reproduksi jamur, terdiri dari spora dan konidia, ada di mana-mana, di udara banyak. Manusia punya barier alamiah yang baik terhadap infeksi jamur. Penyakit jamur bersifat aksidental fenomenon. Barier utama jamur pada jaringan manusia adalah temperatur yang tinggi 37 derajat Celcius dan redoks potensial. Bila jamur bisa menghadapi barier ini, maka terjadi infeksi dan sifat dari jamur akan berubah. Mikosis ada beberapa macam. Yang superficial, jamur menyerang lapisan keratin, lapisan kulit yang mati, sehingga sering tidak ada keluhan. Semua kosmetik kurang baik. Yang cutaneous, mikosis, sampai ke epidermis, disebabkan oleh jamur dermatomikosis, dermatopita. Penyakitnya disebut dermatomikosis. Subcutaneous mikosis karena trauma. Yang sistemik adalah mikosis, masuknya spora terhimpar melalui respirasi ke alveoli, kemudian akan dilawan oleh makropag alveoli. Biasanya sembuh spontan, kembali tidak, jamur akan ikut sirkulasi menjadi sistemik. Mikosis opportunistik karena kekebalan yang menurun pada pasien. Ini referensinya.
This is Eric from Stanford University, and this is the first video in a three-part series on fungal disease, and the topic is the classification of pathologic fungi. The second video in this series will discuss the specific clinical manifestations and diseases of fungal infections, and the last will cover antifungal medications. The specific learning objectives of this video are to describe the general characteristics of fungi, including cellular structure and microscopic appearance, and to list the 5 primary categories of pathologic fungi, including representative species within each. Let me first ask, what exactly is a fungus? In common usage, most laypeople would suggest these organisms, mushrooms. Perhaps some would point out also that mold on an old piece of fruit is also a form of fungus. But the fungal kingdom also includes things that we don't necessarily think about initially as being fungi. For example, you know all that blue stuff in blue cheese? Guess what it is? That's right, fungus, specifically a genus of mold called Penicillium, which fascinatingly is the same mold genus that's responsible for penicillin. In addition, you know that yeast that we use to make bread rise and to brew beer? Also fungus. And even miso paste is produced in part by a species of Aspergillus. So there's a few examples of fungi, but how do we define it more precisely? In the most general sense, a fungus is any organism that is eukaryotic, has a rigid cell wall containing molecules called glucans and chitin, and are incapable of producing food. That is, they, along with animals, are heterotrophs, as opposed to organisms such as plants that can produce their own food from an external, non-organic energy source, and which are called autotrophs. Their cellular structure is actually very important in both defining what a fungus is, as well as understanding the mechanisms used by antifungal medications. First, like all cells, they have a cell membrane composed of a phospholipid bilayer. However, instead of that bilayer incorporating cholesterol, like animals do, it incorporates a different sterol called ergosterol. Just outside of the bilayer exists a thin layer of chitin. If chitin sounds familiar to you, that's because you learned about it in middle school biology as the polymer of N-acetylglucosamine that provides the strength and rigidity to the exoskeleton of insects and crustaceans. Next is a crisscrossing network of glucans, which are polysaccharides composed of glucose monomers. Glucans include glycogen, which animals use to store energy, as well as cellulose, which is found in plant cell walls. In fungal cell walls, the predominant glucan is beta-1,3-glucan, represented here in red. And this is interwoven with other glucans, which vary between fungal species. Finally, anchored within this polysaccharide matrix are various glycoproteins. When we discuss the cell membrane of a fungus, we're referring only to the phospholipid bilayer. The cell wall refers to everything external to the membrane. A question both interesting and instructive comes up once in a while regarding whether fungi are more closely related to plants or animals. The obvious answer to most people is, of course, plants. But let's take a closer look at that comparison. For plants, animal, and fungi, are they unicellular or multicellular? Both plants and animals are always multicellular, but fungi can actually be either. Which of these have a cell wall? Plants and fungi. Does the cell membrane contain sterols? No for plants, but yes to animals and fungi. Do organisms in the kingdoms employ chitin in their biology? No for plants. As mentioned a moment ago, some animals do, and all fungi do. Which of these display locomotion? Neither plants nor fungi do, while almost all animals do. And I already mentioned their respective nutritional models. Plants are autotrophs, producing their own food, while animals and fungi are heterotrophs, which require autotrophs for the production of food. Finally, one more line. Can they infect humans and cause pathologic disease? No for plants, but yes to both animals and fungi. So looking at this chart, it might seem like more of an even split as to whether fungi are more like plants or animals. However, recent genetic analysis has determined that despite the conventional wisdom to the contrary, fungi are more closely related to animals than to plants. I'm going to now start discussing the specific biological categories of pathologic fungi. The most important distinction to make is that between yeast and molds. Yeast are typically unicellular, though some species can occasionally organize into filamentous structures called hyphae and pseudohyphae, of which we'll see a picture in a minute. Molds are multicellular and are typically organized into hyphae. Yeasts are usually white in color. Molds, as you may have personally observed in either your refrigerator or bathroom, can exist in a wide variety of colors. Yeast reproduction is typically by an asexual process called budding. Mold reproduction is typically through spore formation, which can be either sexual or asexual. And finally, yeast forms smooth, round colonies when cultured in the lab, while mold colonies appear fuzzy. The single most clinically relevant fungal group is a yeast genus called Candida. Candida contains multiple species which cause disease in humans. By far the most common is called C. albicans. This is a commensal organism normally present in the oropharynx, GI tract, and vagina. It causes opportunistic infections in immunocompromised states or when the normal bacterial flora is disrupted by, for example, antibiotic use. Candidal infections can show up almost anywhere in the body, as we'll see in the next video. There are 4 other important, though much less common, candidal species. They are glabrata, Krusei, Parapsilosis, and Tropicalis. In some ways, glabrata is the odd one out, as it tends to be physically smaller than the others, and phylogenetic studies indicate that it is more closely related to other non-candidal yeast species. It's very possible that the next decade will see the organisms currently known as Candida glabrata reassigned to a different genus and possibly given a new name. The other medically important yeast genus is Cryptococcus, which also causes opportunistic infections, particularly meningitis and pneumonia. It has 2 species which cause disease in humans. First, the more common of the 2 is Cryptococcus neoformans. This is found in soil samples throughout the world and is most commonly associated with birds, particularly pigeons and chickens, as well as rotting vegetation. The less common species is Cryptococcus gattii, which I believe is the correct pronunciation. This is found predominantly in tropical and subtropical regions, though has just recently emerged in the Pacific Northwest, particularly Vancouver Island. It's believed to be associated with exposure not to animals, but rather to specific tree species. Let's move on to molds. Although they all seem to look more or less about the same macroscopically to the eye, they are actually an extremely diverse group of organisms. The vast majority of them are actually non-pathologic. Most pathologic species fall into 2 specific groups, the genus Aspergillus and the order Mucorales, which includes genera Mucor, Rhizopus, and Rhizomucor. Both mold groups and their spores are found in soil and decaying vegetation throughout the world, and both can cause a particularly aggressive and devastating rhinosinusitis, as well as various forms of pulmonary infection in immunocompromised patients. An important distinguishing characteristic of molds, which deserves a little bit more attention, is their microscopic structure. As mentioned earlier, molds are predominantly composed of hyphae, which are long, filamentous branching structures. Hyphae in some species contain septa, which are observable internal cross walls, while others do not. Another microscopic feature of molds is conidia, which are the clustered spores responsible for most asexual reproduction and are the primary vector for humans to become infected. A cluster of conidia is attached to the hyphae via a stalk called a conidiophore. Different species of mold can be largely identified by their specific morphology of these microscopic structures. For example, compare the upper mold to this one. Notice how the conidiophore-conidia complex looks completely different. That variation between species can be helpful in identifying these in the microlab. So we've talked about yeast, and we've talked about molds. What's left? The next major category are the dimorphic fungi, sometimes referred to as endemic fungi. And what makes them distinct is that they can act as either yeast or molds. They typically exist in the environment as a mold, where they are found in the soil. Then if that soil gets disturbed and the spores are sent up into the air, they can be inhaled into a person's lungs, where they grow as encapsulated yeast. As you might imagine, since the means of human infection by most dimorphic fungi requires inhalation, the most common manifestation are pulmonary infections, particularly subacute ones, which can develop over days to weeks, rather than over hours to days. Something else about the dimorphic fungi, which is why they're sometimes referred to as endemic fungi, is that they're very region-specific. So while Candida and Aspergillus are really found throughout the world, examples of dimorphic fungi are really found concentrated in very specific geographic locations. Some examples include histoplasma, located in the American Midwest and South. This organism is particularly fond of areas frequented by birds and bats. Birds and bats aren't necessarily reservoirs themselves, but their droppings can apparently alter the soil chemistry to make it favorable for histoplasma growth. Other examples include blastomyces, also found in the American Midwest and South, as well as a bit in Canada. Coccidioides, which is a little bit of a tongue twister, and most people refer to as just cocci, of which there are actually two main pathologic species, is found in the American Southwest. Paracoccidioides, located in Central and South America, which is more likely to lead to a much more chronic infection than the other fungi on this list. And Sporothrix schenkii, which is the one exception to the geographic specificity of these organisms because this is found throughout the world. And this is the one dimorphic fungus which is also more commonly acquired via the skin rather than in inhalation through the lungs. The next major category of fungi are the dermatophytes. These are a collection of fungi which all share the ability to metabolize and subsist on keratin, which is the key structural protein in human skin. Dermatophytes cause superficial infections only, and these are typically named after the part of the body involved. For example, a dermatophyte infection of the torso is called tinea corporis, corporis for body, and I'll be discussing all the various dermatophyte infections in the next video. Identification of the exact species is usually unnecessary for diagnosis and treatment, and in fact, I would guess that excluding dermatologists, not one doctor in 100 could even name a single specific dermatophyte species off the top of his or her head. Dermatophyte infections are typically only a cosmetic problem, but they can be surprisingly difficult to eradicate. The last of the pathologic fungi to discuss is Pneumocystis. The taxonomy of Pneumocystis is a source of continued confusion among doctors. It was originally believed to be a protozoan parasite in humans and rats, at which time it was named Pneumocystis carinii. It was determined to actually be a fungus in the 1980s, and the human pathogen was later renamed Pneumocystis urobetsii once it was evident that it was a separate species from the rat pathogen. For historical reasons related to nomenclature battles, urobetsii is frequently yet probably technically incorrectly spelled with just one I at the very end. However, people still refer to this as Pneumocystis carinii not infrequently. Partly this has been perpetuated by the insistence of holding on to the acronym PCP. PCP is the name given to a clinically distinct form of pneumonia caused by Pneumocystis. It originally stood for Pneumocystis carinii pneumonia, but even after the species was renamed, clinicians did not want to change the name of the disease, so now they claim it stands for just Pneumocystis pneumonia, which in my opinion is just a bit silly. Another relevant characteristic of Pneumocystis is that because of differences in biochemistry and cellular structure compared to other fungi, Pneumocystis infections are treated with antibacterial antibiotics and not antifungals, which further contributes to the confusion regarding taxonomy. In addition to the major types of pathologic fungi, there are some bacteria which masquerade as fungi, specifically Nocardia and Actinomyces. Both of these develop filamentous branching structures visible with microscopy, and they cause subacute and chronic infections of the lungs, central nervous system, and soft tissue, most commonly seen in immunocompromised patients. Infections by Nocardia and Actinomyces can be clinically indistinguishable from fungal disease, except for the fact that, as with Pneumocystis, they are only susceptible to antibacterial antibiotics and not to antifungals. So that's it. To quickly summarize, there are 5 major clinical categories of pathologic fungi. The yeast, which include Candida, of which C. albicans is the most common species, and Cryptococcus. The molds, which include Aspergillus and Mucorales. The dimorphic fungi, including Histoplasma, Blastomyces, Coccidioides, Paracoccidioides, and Sporothrix. The dermatophytes, the individual species of which do not matter. And finally, Pneumocystis, a single species which gets its own category because it's quite clinically distinct from the other fungi listed here. And please note that this classification scheme is more of a practical scheme based on their clinical manifestations and is not meant to be an accurate representation of their formal taxonomy. I hope you enjoyed the video. The next in this short series will cover the various clinical manifestations of fungal infections.